Okay, so we continue where we left off. Okay, so the next point which is important is the difference between the inorganic phosphate. Usually people will draw it as Pi, and also the phosphate from ATP. Inorganic phosphate, as you can see from I think reaction number somewhere reaction number 7, is actually the phosphate that is actually floating around in the cells. But the phosphate that is bound to ATP, we usually not call them as inorganic phosphate. So when I told you guys that ADP plus Pi equivalent to ATP, usually this adenosine diphosphate will combine together with inorganic phosphate floating in the cell and becomes ATP. Okay, so that's the difference. Next, the point is the two difference between two enzymes, which both of them uh, have the same function, which is hexokinase and glucokinase. As you can see from here, this is hexokinase. Means that this enzyme, they are actually in all the other cells in the body except for liver. But in liver, instead of hexokinase, it becomes glucokinase. Okay, so if you are hardworking enough, you can try find why both of these enzymes, they have the same function, but in different location they can be different. Okay, for example, why in the muscle contains hexokinase but why in the liver contains glucokinase. Okay, you can try ask me if you don't find the answer. Okay, and you need to remember that this glycolysis reaction only happens in cytoplasm, so no oxygen is needed. If this pyruvate need to is if this pyruvate need to be converted, needs to enter into the mitochondria, they must have oxygen. Pay attention at reaction 5. Glucose breaks into 2. Okay, as you can see from here, glucose breaks into 2. So you need 2 times 2 for everything that you get after this reaction. So the investment is 2 ATP, but the final product is actually 4 ATP, but we minus by 2, we get 2 pyruvate and 2 NADH. Okay, so you will have 2 pyruvate here. Okay, you need to put 2 I think here. Okay, so like I told you before, the most important part in this glycolysis is regulator points. And if you can see from this figure, they just show you actually the regulator points. So glucose to glucose 6 phosphate is the first reaction, which is the regulator points, reaction number 1. And then fructose 6 phosphate to fructose 6 biphosphate is actually the reaction number 3, which is the most important regulator points. And then the final reaction is phosphoenolpyruvate to pyruvate, which is the number 10th reaction, the third regulator points. And as you can see from here, usually the inhibitor shown in red is usually the ATP and citrate, while the activator is usually ADP and AMP. ADP stands for adenosine diphosphate and AMP stands for adenosine monophosphate 1-phosphate. ADP is 2-phosphate, means that the cell is starved of energy. They don't have enough energy. So what they do is they will stimulate the process of glycolysis. But if the cell contains lots of ATP, triphosphate, the complete form of energy molecule, and also citrate, it shows that the cell is actually already have enough energy. So they will inhibit the regulator points. Okay, these are the two most common ones. Other common activator inhibitor is, for the activator, it's going to be NAD plus or FAD, but for the inhibitor it's going to be NADH and FADH2, because it shows you that the cell already full of energy in the form of NADH, so you don't have to stimulate the glycolysis anymore. Okay, citrate, you can see the citrate here, but the citrate can be a little bit weird, okay, because citrate is actually one of the molecule inside the T6 cycle. So that's why sometimes the T6 cycle called as citric acid cycle, because the presence of citrate here. So if we already have lots of citrate here, it actually shows that the cell is already full of energy, so it will slow down the glycolysis reaction. Other inhibitor can be the product of the reaction. For example, glucose to glucose 6-phosphate. The product is glucose 6-phosphate, right? So this glucose 6-phosphate can actually go back to hexokinase here and inhibit the reaction, because it tells you that the cell is already full of glucose 6-phosphate. Okay, so you don't have to produce anymore. You don't have to break the glucose anymore. So it can inhibit its own enzyme. This reaction we call as feedback inhibition. Okay, like I told you, the reaction 3 is the most important, but reaction 1 and 10 is also the regulator points, but not as important as reaction number 3. The characteristic, as I told you before, is big change in negative energy, the delta G, irreversible, and reverse reaction requires different path of enzyme. Okay, why ATP and ADH always inhibits while ADP or NP always activate, like I told you before. Alright, this shows you whether the cell got enough energy or not. Inhibition by products through allosteric site is known as feedback inhibition. Allosteric site means that this is the site of enzyme that is different from the active site. Okay, so if you get you got an enzyme that got the active site circle in shape, you will have the active site here, but the allosteric site is different from the uh, active site. Okay, binding of activator or inhibitor at the allosteric site can either stimulate or inhibit the reaction. You can also have non-enzymatic regulation through hormone, for example, insulin, glucagon, and so on. Okay, and there is one case study which is anemia in person with defective pyruvate kinase. Okay, the pyruvate kinase is actually an enzyme in reaction number 10. Okay, from phosphoenopyruvate, it will produce pyruvate through pyruvate kinase. So in red blood cell, certain red blood cell will have the deficiency in pyruvate kinase, means that they cannot produce enough ATP, because this reaction also produce one ATP, right? So what happens is the cell don't have enough energy and the shape can be distorted. Because the shape is distorted, the body will destroy the red blood cell, and that person will get anemia. So there will be certain treatment. Okay, if possible, you can try find it in the Google. If you cannot find it, you can refer to the other lecture notes, or you can actually find it in the Google, Wikipedia. Okay, the next important part is about the fates of pyruvate. Means what happens to the pyruvate after this reaction. Okay, from this figure, you can see there are actually 4 different fates. The first one is what we call, they can be divided into 2. The first is aerobic fate, the second is anaerobic fate. The aerobic fate, it can produce gluconeogenesis in the liver by producing a molecule we call as oxaloacetate. Oxaloacetate is also one of the intermediate inside the TCA cycle. Okay, the second fate is also under aerobic condition, where your cell is fat, means that you are not under fasting condition. Okay, so if fasting, of course you need to produce glucose, right? But if you don't have to produce glucose, it will enter into mitochondria and produce energy through TCA cycle. So the second fate is actually the production of acetyl-CoA. The third fate, which is anaerobic condition, is the lactic acid production. This can happen in both human and also microorganism. So I think you've heard about the lactic acid production, right, in lactic acid bacteria, right? So you need to add bacteria here, and this lactic acid will eventually be converted back into glucose in the liver. The last phase is actually happens only in the microorganism, which is the ethanol production. So although this microorganism, they have oxygen, they still prefer to not use the oxygen and produce the ethanol. This can be seen in yeast and bacteria, for example, in the beer production. Okay, we take a look at this point. Okay, there are 2 phases, divided into 2 phases. The first one is aerobic and anaerobic. The product is acetyl-CoA for normal aerobic, also the acetate for normal condition, but under fasting condition. If you don't have enough food, enough glucose, the liver will convert it in. So this one only happens in the liver. This one happens in all cells. And then the third one is lactic acid under not enough oxygen. or anaerobic condition, and ethanol in microbes. Okay, one of the question you can ponder is the bread fermented by yeast. So which one is the fate of the pyruvate? Okay, is ethanol, because it means that we are eating bread with ethanol. But why won't we get drunk when we eat the bread? Okay, you can think about it. Okay, location. If the aerobic respiration, the pyruvate must enter into mitochondria, into pyruvate dehydrogenase complex. We will take a look at this during the TCA cycle. And if anaerobic, it will stay only in the cytoplasm. It won't enter into mitochondria. Okay, and in human, it will turn into lactic acid. Okay, later case study explain how lactic acid can form during exercise and how alcohol can disrupt it. Okay, so this one we can take a look later in the lecture of gluconeogenesis, so you can understand better what happens. Okay, so this slide is nothing much. It's just want to show you that actually when we eat, we will eat lots of a different kind of sugar, right? For example, we will eat fructose, we will eat galactose in the form of milk, we also take glucose. Okay, so if we just eat glucose, the reaction is simple. It starts from glucose and it will go through the normal glycolysis. But if we eat other type of sugar, for example fructose will undergo one reaction first in the liver, and then it will convert it into the product in the middle of glycolysis. But if we take milk, for example galactose, this galactose must undergo certain reaction, I think 5 reaction first before it can enter into the glycolysis. Okay, so eventually everything will merge into glycolysis. That's why we call catabolism as converging reaction. Okay, like I told you in the previous lecture. Okay, so although you can see different kind of sugar here, for example galactose, glucose, fructose, and all of them bring the same amount of energy, which is 4 kilocalories. Okay, you will learn about this in nutrition with Dr. Shazini later on, and Dr. Nida. All of this sugar actually they have what we call as different physiological effect to the body. Okay, so for example, if you eat more fructose, you can get fatty liver, or fructose actually can be taken by the diabetic person. But if you eat galactose, people say galactose is the healthiest sugar out of 3. So they are reason because they undergo different reaction before they can enter into the glycolytic pathway. Okay, you can try go through this case study how to manipulate sugar intake for the diabetic patient, okay, to understand how different sugar actually affect your body. Okay, so this is a tutorial. I won't answer it for you. You can try answer it yourself. But if you don't know anything, you can always WhatsApp or email me. Okay, so I will try to answer it for you through discussion. Okay, until then, thank you. I'll see you in the next lecture.